All right. Well, Amy, Proal, I'm so glad that you were able to join us this afternoon to answer some of our viewers' questions about stealth infection. And wanted to start off by asking you if you wouldn't mind telling us a little bit about maybe anything in your personal past that led you into your interest and passion about exploring microbes.
Sure. Well, thanks, first of all, Wally, thanks for having me. This is a cool opportunity to talk to people more about what I spend a lot of time thinking about and writing about. Yeah, my own life and my own medical history actually played a lot into how I began to be interested in basically studying microbes and the microbiome, which are the communities of microbes that live in and on us, right? So that story began mostly when I was at Georgetown University. I was an undergraduate. I was a pre-med student, so I was already, studying bio. But and I was doing research for a thesis on rats eating a ketogenic diet. It was all very interesting. And then I started to feel sick myself. In like ways that were just very difficult. I had headaches. I always felt like I had a cold. When I look back at my medical records, I would just go to the Georgetown, Health Student Center, which is in retrospect a very bad place to have gone because they kind of just deal with mono or something. I would just go there and be like, I have a cold, like flu stuff. And then I had difficulty sleeping. And, after a while, it was like, okay, this doesn't seem normal. But when I would see a doctor, this is typical with the diagnosis I eventually got, which was ME, CFS. I think ME is the name now that everyone in the community prefers. Some people know it as chronic fatigue syndrome. It's a changing name. And I also got eventually a diagnosis of fibromyalgia. So kind of this mix of malaise and pain and a lot of stuff. But at the time, and still now, unfortunately, those illnesses are not well recognized or treated. So I went through years of seeing doctors that just told me that my symptoms were in my head or that I had some other kind of problem. So I was sent to a ton of psychiatrists. So under those, and then you need just to be there, sitting with the psychiatrist. And luckily, I was pretty confident in who I was as a person and my ability to do stuff. And I knew that these were very concrete physical, it was like pain, flu-like, I didn't even think for a moment that it was anything psychiatric, but getting other people to think that was a real challenge. So I went through all these years of just being basically ignored. And so I kind of took matters into my own hands. I mean, I don't think, I didn't know what else to do. So I got on Google and started looking, up and first basically kind of diagnosed myself. First even found out that chronic fatigue syndrome, ME, or all this stuff was even a thing, right? And then when I went in that direction, I think because the symptoms at that time, the description for what was called chronic fatigue syndrome really matched what I was feeling. I was like, all right, let me run with this. And when I started looking at the history of that illness, there's a huge correlation between infection and the illness. And this is still a matter of debate today, but most people who get sick have at least some sort of infectious trigger, or, I actually don't think trigger is a good word. I think trigger, the idea, because there's still debate now there in the community that, a pathogen might come in and trigger symptoms and then be eradicated. I actually doubt that's the case. I think in most of these cases, people get sick with an increasing number of infection, and the infection will probably remain, whatever the microbe or pathogen is, probably remains in the person's body as part of their microbiome community. It's kind of causing trouble. So I was actually, yeah.
Perhaps like a combination of infections then.
Yeah, exactly. And I could get more into that, but I don't, I think this kind of precipitating, infection that's associated with chronic fatigue syndrome, ME, CFS is, I think there's a lot of different exposures that lead up to a point where symptoms become more obvious. So I don't necessarily only think about the, one exposure or something as being important. But long story short, when I was looking at the cases of other people who had this, there was always some tie back to having had some sort of very flu-like illness when things got worse, or some sort of this, or even just some sort of environmental exposure to mold, something, something that seemed to throw the immune system more off and, and have some sort of, and then lead to symptoms that were very flu-like and sort of chronically infectious in nature. Right. Sure. So I was definitely running off the way I felt myself, which was that I felt like I had a raging chronic infection. So with that in mind, this all started making sense. So I began to look at, infection, chronic infection more, and in order to do that at the time, this was around 2003, 2004. So there was no real concept at that time of the human microbiome, which I can go into more, which is basically the sum of microbes that live on and in us. And we now know there are trillions of these microbes that persist in and on us. And so, but that at that time was barely known. There were a couple studies. I remember reading a study by Dave Relman, who's a researcher at Stanford, definitely one of my earliest kind of role models. And he had used early kind of DNA sequencing tools to look for microbes in human blood. And he came back, he looked at healthy human blood, and he was like, there's bacteria here, right? And so I would see that, and I was like, wait a second, like when you're using these new kind of tests that are able to better, detect these bacteria, which were basically tests that were looking at their DNA or their genes rather than just whether they could grow in a petri dish or, show up on a test, it seemed like there were more microbes that were capable of persisting in the body. And so going down that, I actually began to look more at, interestingly, researchers who had studied, at that time, what were called stealth pathogens in basically the 40s, 50s, 60s, at that time, it seems there was actually much more interest in microbes and the different states that they could persist in and chronic persistence. And those years and even earlier years than there was later on, kind of around the 80s and 90s, when the theory of autoimmunity really gained hold, which is a theory that basically part of it posits that the body's mostly sterile, doesn't involve any kind of microbes in these inflammatory conditions. When that theory came in the 80s, it seems to have really curtailed a lot of this early microbiology work that was looking more closely at infectious agents capable of persisting in the human body. So I kind of went back, and looked at it.
So was that also a time when there was a lot of focus on the whole concept of immune tolerance, and that these were problems with abnormalities of that mechanism of immune tolerance?
Right. So I would run up against this theory of autoimmunity, which, sometimes was even postulated to be happening with ME/CFS that I had. And the working model with that is that the immune system, kind of for reasons that are very unclear to me, that's one part of the theory that doesn't really hold up in my mind. The immune system loses what's called tolerance, which is sort of the ability to manage, correctly manage what it's doing, and begins to sort of attack its own tissues. Like the body will begin, the immune system will essentially attack self. And so I think what's key to understand about that theory is that theory was developed during a time when the consensus among the scientific community said that the body was essentially sterile. So this work that I went and looked back at researchers who were looking at kind of stealth infection and microbes that could persist in the body was always a little bit sidelined in the microbiology and medical world. In fact, a lot of these researchers who I think did amazing work, they really got no credit for it. In fact, they ended up almost being very marginalized. So
I don't think I've even heard of most of those people.
Yeah, there are some amazing people, and even, some amazing women who just did. This one woman, Virginia Livingston, she honestly, she published textbooks and other books, and, the photographs she took of these. And one of the issues at the time that I think was difficult is that these early researchers looking at microbes in the body were always faced with the, kind of stuff was levied against their work that contamination was playing a role, because they would take a human sample, they would try to take human tissue, look for microbes in that sample. When I look at the methodologies they were using, I really think that most of the time they were actually getting microbes from that sample. But what a lot of their detractors would say was like, oh no, that microbe was probably in the lab, or that microbe. So it was very hard for them to prove that the microbes that they were studying from in human tissue didn't come from another source. And so they kind of faced this all the time. If someone wanted to sort of dismiss their work, they would just say, oh, contamination.
They'll be able to use that argument for a long time yet.
Right. And it's still something I see sometimes, because right now, for example, there was a recent brain microbiome study that looked at, they took, I'm pretty sure, brains removed during an autopsy, and they, started, using this time newer tools that look for microbes based on their DNA, not just, sort of their presence and ability to grow in the lab. But even with those tools, the researchers who did this study went through a huge amount of, and which I thought was reasonable, a lot of experiments to try to prove that this was not contamination. It was a major issue they already knew that they were going to face. I think anytime you're really looking for microbes in a new environment, like the brain.
Especially the brain.
Exactly, which is, in medical school everywhere taught to be at this point sterile. You really have to, cover your tracks and make sure it's going to be hard enough to accept as a new paradigm. So I did notice that even now, you have to be very careful to make sure. And I think that's fair. I don't think it's not, I think it's a good idea to make sure that your samples aren't contaminated. I don't think that that's bad to do, but that's
not what we're after. Yeah, exactly.
Yeah. So with these earlier researchers, and I actually just went ahead and kind of called some of them. Like this one guy named Gerald Deming, he worked at Tulane for decades studying, again, what at that time were kind of more called stealth pathogens, which at this time are just members of the human microbiome. Like we don't even have to refer to them as stealth anymore. See, that's what I think right now, since we now understand that now we really actually accept the fact that so many microbes live in and on the human body. They're just part of our microbiome communities. There's nothing stealth about these microbes. I mean, they're just there. Of course, at the time, this was considered, a little on the fringe. So then they were stealth, So he was really cool. I found him, he was living in Switzerland, and I basically just did what you're doing. I was like, hey, can we talk? And we Skyped, and, he walked me through more of what he had done back in the day. And he had done some amazing work looking at, people with all kinds of conditions, chronic kidney disease, other issues, bladder type disorders that are really associated with infection. And he found this wide range, especially looking at bacteria, because bacteria were the easiest, at that time to look at. So it was mostly a focus on bacteria, but, he really had found a lot of stuff. And speaking to him, I could see what a thorough, intelligent researcher he was. And so, all of a sudden I was like, wait, because as a patient, I was going to these doctors and they were telling me there's nothing wrong with me. And they would run a, standard blood panel that looks for, 2 pathogens or something like that, Epstein-Barr or something, and then they would tell me, there's no way that there could be infection affecting your condition. And
culture negative.
Pretty soon I was like, well, there are a lot of ways infection could be affecting this condition. It's just that our tests are not picking up on it. And whatever work has been done on microbes really hasn't reached the medical community. I mean, I could just see these immediate disparities between what I was being told in the clinic because, the information hadn't caught up then and, what I was finding online. And so I was really lucky at that point to have a really great mentor at Georgetown who allowed me to switch my thesis work from before working on the rats with the ketogenic diet towards just writing a literature-based thesis about chronic fatigue syndrome. So he just let me, and it was also very good since I really couldn't get up anymore. I wrote that from my bed and just did like an investigation at the time of what could be found on it. And then that was my thesis that got me into stuff. And while I was doing that, I did come across the work of Trevor Marshall, who I think you'll probably ask me about, because I ended up becoming very interested in the work of this, again, researcher named Trevor Marshall, who's still one of my closest colleagues and friends, to be honest.
Why don't we just segue into that? So
Yeah.
you were pretty sick. You had to kind of change your educational path a little bit.
Yeah, enough.
Because of that. And then you ran across the work of Dr. Marshall.
Mm-hmm.
And tell us a little bit about your association with him and where that led your career. Right.
So it's interesting because I had obviously had a very strict scientific training at Georgetown. And so, in theory, you're supposed to wait until there's a randomized placebo-controlled trial on any treatment you do. And I came from that mindset, but then as a patient who couldn't move anymore or get out of bed, I realized that I was just going to have to try something that was more experimental. I just was not going to sit there and do nothing, and nothing else had been offered to me. So I found Trevor Marshall, who at that time had created what became known as the Marshall Protocol. He never actually named it that, like people just started calling it that. And it was based on what really made sense to me and still makes a huge amount of sense to me theoretically at first, which was that no, the immune system isn't attacking itself or going crazy or anything in most of these conditions. What's really happening is that there are more microbes in the body than we usually give credit for. At this point, again, the microbiome was still not very well characterized or understood. And it's a lot of these chronic illnesses may be in large part due to chronic pathogens able to persist in the body that are able to dysregulate human metabolism, dysregulate all kinds of, human pathways, and essentially cause inflammation and, all kinds of issues just by, persisting chronically in the body, right? And again, combining that with all this research I'd done on all these microbes, all these researchers who had found that kind of thing, it made a lot of sense. So what his protocol basically encouraged was to activate the immune system, as opposed to most conventional treatments even today, which shut down the immune system in an effort to basically control symptoms. He was saying, wait, if there's chronic microbes or pathogens that might be, at the root cause of what is causing the symptoms, what we should really do to the immune system is support it or activate it. If we do that, maybe the immune system itself, which has obviously evolved with these microbes, since the beginning of time. So maybe we can almost use the body's own immune defenses, as long as we're able to support them, to kind of figure out more what's going on and allow the immune system itself to better manage the problem and possibly kill some of these chronic infectious agents and allow people to move to a place where they feel better. And I was like, that makes a lot of sense. That's something I want to try to do, because I should add with my own story, which I think adds to my interest in microbes, is that when I was young, I still don't have much of an explanation for why this happened. When I was young, and I have a twin sister, a fraternal twin sister. So we're fraternal, so it's not a perfect comparison. When I was 4, I got viral meningitis, and it was a really severe case. And I remember almost being almost hospitalized, I almost died, whatever. I was treated with a huge number of steroids, strangely antibiotics, stuff like that, even though it was viral. And then after that, even though I had been vaccinated for measles, mumps, rubella, I got measles, mumps, and rubella on 3 separate occasions. And I just don't know why. I don't know if they didn't put the needle in my arm. I don't know if my immune system there was something about the meningitis that screwed up, which is actually very possible now, looking more at, how these pathogens, usually when you get a serious infection with one pathogen and it can kind of stick around, it will adjust the immune system in a way that benefits its survival. So it's not impossible to say that serious infection and disease didn't kind of, allow my immune system to get these other conditions. And then I had a case of pneumonia that was really crazy, scarlet fever, which I don't even know if anyone else gets. And I think that's it. But it was this huge, progression of illnesses that I was just hospitalized for. And then around the age of 7, I came out of it, and I was totally fine, it seemed, at that point. And I had always seemed very healthy. And then I went on to be like a star athlete, all this stuff. And it wasn't until I was like 20 or 21 that I started to get all these chronic symptoms. And again, when I'm trying to think well, what did I do? I knew there was nothing that I had done, really. I had lived a pretty healthy, good life that compared to everyone else's that wasn't getting sick. So I went back and said, I wonder if any of those, severe infectious illnesses had any, impact on what I have. And so again, that became very interesting to me, because I looked at, even back at those infections and can see that, in my opinion, it's definitely possible that some of the pathogens that cause those things actually stuck around in some different chronic forms.
That's really interesting. So your personal story actually goes back to about age 4, really.
It really does, yeah. And you can see all these cues that keep pointing me towards infection. And so that's why I just, kept going in that direction. But anyway, so when I found this Marshall protocol that said, hey, let's support your immune system to see if we can address chronic or, infection, I was like, yeah, I think this is what I should be doing. I should be trying to do this. And what's interesting about the treatment, and I think patients get this more than physicians sometimes, is that there's basically a Herxheimer response. And I think patients who have your chronic Lyme stuff or things are used to this concept of Herxheimer. It depends, kind of how you've been treated, which is basically a phenomenon in which if the immune system on its own actually successfully targets a pathogen, symptoms will actually go up for a while. The symptoms related to what that pathogen might have been, causing, in the sense that in order for the immune system to kill the pathogen, there has to be, an immune reaction towards that microbe. Sometimes when I'm describing that to people, I'll just ask them what they think happens when they get the flu, right? When you get the flu isn't caused by the virus just eating your cells or something. What your symptoms really are is your immune system, trying to battle the virus that causes the flu. And that battle between the microbe and the immune system is what causes, inflammatory cytokines or molecules to be released on the part of the immune system. And then if the pathogen dies, there's toxin release, and there's a lot of times debris from infected cells, because a lot of these pathogens persist inside the cells of the immune system. So you kind of get this toxic mess that the immune system has to deal with. There's inflammation in order for that to happen in the first place. So if you actually successfully kill a pathogen under these conditions, you would expect your symptoms to actually go up, but then maybe come back down if you actually successfully eradicate the pathogen. And that's really what
ask you now, I think that, correct me if I'm wrong, but that was Dr. Herxheimer, who was like a German microbiologist, and that was back when they were treating syphilis, I believe.
That was, I think, the first example of where they really saw this reaction. Yeah.
And I think they were even using potentially toxic metals at some point, like bismuth.
I think they were using exactly mercury. And so, I don't know, I have no idea what the, kind of effects of using that are, but what would happen is in some way this mercury, and I'm not sure I even get the mechanisms, was able to, it seems, stimulate the immune system or actually just cause a reaction towards, the syphilis. And patients would get very, they would be treated aggressively and get very, 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 very sick while the mercury was kind of, I guess, causing the immune system to combat
I suspect that you're right, that it was actually killing some of the pathogens, and they were breaking down, or the cells that were housing them.
Right.
You're getting like a cytokine storm from all of the inflammatory action.
Now, I think a good example of this, actually, that people don't necessarily correlate with Herxheimer, but I do, is cancer immunotherapy. If you look at some of these new immunotherapies that are being developed for cancer, they'll, for example, take a patient's T cells or something and engineer them in a way that causes them to be more active and, in my opinion, probably target, more pathogens that might be involved in the cancer. That's debatable.
I was going to say, I would love to get you started someday on talking about the role of infection in cancer.
I think there's no way that, the microbiome or pathogens in the microbiome don't play at least some role in most cancers. When we look at colon cancer now, for example, it's totally tied to microbiome. So if you look at that, as sort of a beginning point, because it's easy, not easy, but at least in the colon, you can get fecal samples. You can is it easier to study the microbes in the colon than it is to, let's say, study the microbes in the blood or the lungs at the moment. So these studies that have tied colon cancer to changes in microbiome composition and to pathogens in the microbiome, there's quite a few right now. And I think you would have trouble almost arguing with anyone that the microbiome being out of balance doesn't contribute or drive parts of at least the colon cancer process and even tumor formation, right? So looking at that, I think it's of great interest to me that when patients with cancer undergo immunotherapy, where their immune systems are actually, again, activated towards the cancer, that, they have this similar cytokine storm reaction where, you could say, well, it could just be the immune system going, getting very excited or whatever, but that it'll be such a profound cytokine reaction as the, immunotherapy treatment tries to get at the tumor or the cancer, that some people actually have, died from this. It's an incredibly severe reaction right now. So I do think that you see this trend where if you activate the immune system towards what I would say would be infection, or even just, dysregulation or imbalance or tumors, whenever the immune system has to really address a problem, you will see an increase in symptoms as that happens. And
that's in part because some of the effectors or inflammatory mediators that the immune system uses have actually quite a toxic effect against our own cells and tissues. Isn't that right?
Yeah. I mean, in what sense?
Well, for example, like histamine release,
right?
Is equally toxic to us.
Yeah, exactly.
It would be to a potential pathogen or
Right.
bad
Well,
guy.
there's definitely probably issues where if you accumulate enough of a chronic infectious burden, which in my mind now is just, having microbiome populations that are probably not in a state of balance and that have more active pathogens that are acting more in a way that moves in almost the entire community towards virulence or actually being able to, promote and cause disease, then yeah, you're going to have, the inflammation, your immune system. I think there's at a point like stalemates that sort of develop. I mean, that's what it seems like, where the immune system is probably trying to, improve what's going on there and try to target pathogens. But if it's too hard, you might get this chronic stalemate at that point. There's a lot of issues. Now we know so I'll just clarify again with the microbiome, there's these trillions of organisms that we now persist in every body site. I think right now there's analysis that have looked at everything from the placenta to the amniotic fluid, definitely the blood. So no part of the human body is sterile. We have these communities of microbes that are living in and on us. And it's clearly capable to be in a state of great health and still have these microbe communities. So we know that we can coexist with these microbiome communities. But when, we can also see a lot of ways in which they can begin to move as communities almost towards states in which pathogens begin to take over, in which, more, I would say, innocuous microbes that don't tend to cause disease get outcompeted by those that do. And you tend to see almost one of the biggest trends is lower diversity. As the microbiome moves away from what would be considered a state of health, you'll tend to see, and this is really common now with when people are analyzing the gut microbiome, all the microbes in the gut, is that in a huge number of inflammatory conditions now, they're correlated with what's called dysbiosis or imbalance of the gut microbiome. And in those cases, one of the biggest trends is just lower diversity of species. So it appears that certain pathogens, which I'll usually refer to as keystone pathogens, have kind of, either altered their gene activity and what they're doing to move that community away from balance, or a person maybe has acquired other pathogens that have come in there and started to mix things up in a way in which their survival is being prioritized, and maybe some of these other ones that were, better keeping things in a state of balance are being eliminated or, kept out. And you tend to see this like lack of diversity and stuff like that. So basically, that's a huge trend right now. So going back to that, I think where I was going with this is, if you have all these trillions of microbes in the body, a lot of which can move towards, being pathogens or acting in ways that can promote disease, they're always creating their own proteins and metabolites. So they're always like, they're doing their thing. They especially, these are mostly the bacteria, but viruses as well. They, as a result of their gene expression and their activity, they're always creating, expressing proteins, metabolites. And a lot of those, think how many there are. I mean, if there's trillions of microbes in and on us, there's this constant then, surge throughout our bodies of these, theoretically foreign proteins and metabolites.
I think I've heard somewhere that the genetic diversity of our microbiome probably is about tenfold or more higher than our own personal DNA genetic diversity. So in other words, they have the capability to form all kinds of different proteins and molecules that we can't form.
That is true. There's, our human genome has approximately 21,000 genes, right? So as human, basically as humans, our processes are tied to the microbes within us. But if you try to separate it out, you have, about 21,000 human genes expressing human gene products and metabolites all the time. And then you have these trillions of microbial cells that are expressing their own proteins and metabolites. So by definition, they're more, microbial, or in theory foreign, it depends how you want to call them, proteins and metabolites in the body than there are human at any given point, right? Sure. And there's also just only so many structures that these molecules can have, right? So if you think about it, a lot of what I think, I had a really good training I had good molecular biology kind of training when I was learning about this stuff. So I picture a lot of things in terms of shape and conformation. But if you have microbial metabolites or proteins that are similar in shape to those that are expressed by the human body and the human genome when it's doing its thing, you can see how over time, if the microbes that are, creating proteins and metabolites in the body are actually pathogens, are actually acting more like pathogens, then you can see this opportunity for the human immune system and human pathways to begin to kind of get confused as to which metabolite is coming from where. And so basically a metabolite that might have been created by a pathogen could probably come in and impact a human receptor that was expecting to have a human molecule, sort of adjust its activity. But really now you have a microbial metabolite with a really similar size and shape that's able to bind into that same receptor and alter its activity. So that's where things get really crazy, as in you can actually have microbes and their gene products come in and directly modulate the expression of human pathways. So that in and of itself is an incredible mechanism by which pathogens can cause disease. But then there's almost like this second layer where the proteins and metabolites, because they're similar to human ones as well, the immune system can get confused. And I think this plays a little into the theory of autoimmunity. There probably does begin to be some what's called molecular mimicry, where the human immune system is getting confused between what's human, what's microbe, as things progress towards disease. And it is possible that the immune system could, try to target a molecule that was created by a pathogen and target human tissue instead, because that tissue or something about that tissue had something in similar size and structure to the microbe, to the metabolite from the pathogen that it was originally trying to target. So there could be this, misfiring in some cases where things are not, as things get more and more confused.
Like,
and so,
for example, to
right.
explain, say, thyroid autoantibodies
Yeah.
or something of that nature.
Yeah, I think that's really likely, in that it would make more sense. It just, I know that it makes more sense to me that antibodies, which are what really get caught up in a lot of this, which are molecules that, are created in response to pathogens. The immune system tries to use antibodies to better identify what pathogens to target. So if there's a pathogen, the immune system will try to create an antibody against it. But that antibody, if it's, kind of what's called, has a polyspecific size, it's not very specific for size and shape. Again, if there's a human, protein or whatever that has something similar, that the immune system may begin to target that similar shaped human protein in addition to trying to target the one made by the pathogen. So you get this kind of cross reaction, right? That's definitely possible. But what's most important is that in that scenario, the antibody was originally created in response to a pathogen. So if we want to get at the root cause of what's happening, we would probably want to address that pathogen or the infection instead of using treatments that kind of try to shut down antibody production, which is what we do now.
Sure. Now, to kind of seg into a little bit of different area, I know you have a lot of interest in this whole concept of microbiome communities and the concept of biofilms. And I know some of your recent work has had to do with communication between different species of organisms, pathogens, if you will, or even good guys in some cases. But talk a little bit about that, and especially as it applies to human disease.
Right. Well, see, I think, I started again looking at the microbiome in the early days when we were tracking these early studies that looked at just bacteria usually. And when, we'd look at use DNA technology to look at a sample and we'd say, oh my gosh, there's more bacteria than we thought here. We started to realize there were a lot of bacteria, but over time, these studies were turning up viruses as well, a huge number of viruses. I mean, when I was first looking at the microbiome, it was very bacteria centric. And then all of a sudden, we started to realize that there were all these viral reads coming out of these studies too. And now I think the number of viruses estimated to persist in the microbiome might even be higher than bacteria. That's certainly the case for bacteriophages, which this all gets complicated. Bacteriophages are viruses that infect bacteria. So in any microbiome community, you have bacteria, you also have viruses that actually prey on and infect those bacteria, which have got to be playing a very important role in how those communities are regulated. We really have only touched the edge of studying them though. Literally at this point, I think just last month, a study came out just even, indicating how many there even are. This was a study that actually estimated that 30 million bacteriophages traffic in and out of the human body every day, which is even for me a little inconceivable, right? So millions of these species coming in and out, and we really don't even know what they're doing, which is, of course, the most important part. We need to better understand their activity. We need to better understand how their relationships with bacteria. At any cost, though, or any case, the microbiome is, I think, far more complex as a community. When I refer to the microbiome, I'm just going to go ahead. What I mean is all the microbes that are living in and on us. A lot of people will kind of be referring just to the gut, because that's where the microbiome is best characterized and studied. But at this point, it looks like we're finding microbes everywhere. So when I mean microbiome, I just mean like all these microbes in pretty much all tissue and blood. So these, I think maybe in the early days, there was almost a little bit of hope that this was going to be sort of easy to characterize. Like, oh, look, these are the bacteria and viruses that are in a healthy person. These are the ones that are in a sick person. All we have to do to get, person from sick to healthy is to move from this mix to that mix. And that just didn't work out. There were studies early on that tried to look for what's called a core microbiome, which would be this idea that healthy people might have a certain, core of microbes that were actually in a shape that promotes health. And those studies haven't even really been able to hold up because it seems that this is a very personalized thing.
Isn't this where some of the
person has their own unique mix of microbes?
Even some of the work on fecal transplants, kind of going back to some of that concept.
Right. Well, fecal transplants are basically, a technique that people are using to try to basically change up the microbes in their colon and gut and, GI area. And it's pretty basic. You take a stool sample from a technically what's called a healthy donor, which is someone that just basically doesn't seem to have what you have, or just seems to be in good shape. And if you do it responsibly, you really try to test those samples as much as possible to make sure there aren't any key pathogens in that. There are centers that do that, which I would encourage anyone to go to. Although, to be fair, a lot of people are just doing this in their own homes. A lot. Like, they will just get a fecal sample from a neighbor or someone who seems healthy. They have, illness or GI problems or anything, and they'll basically try to enema those fecal samples from this healthier person into their colon, into their gut, with the goal of switching up the microbes, trying to hope that the healthy person's microbes come in and begin to take hold and sort of colonize over the mix of theoretically kind of unbalanced, unhealthy microbes that they may be harboring. And there have been some pretty impressive success stories of people doing this right now, especially for C. difficile, which is a gut infection that's no small issue. It does seem that bringing in this, healthier, more balanced mix of microbes allows the community to sort of, take down C. difficile as a pathogen, which is really interesting. Again, we're getting into community here. It really isn't, there's, at this point, everything that I've looked at lately is microbes acting together in ways that either lead towards health or disease, or, affect some process. And that goes back to your original question about biofilms and stuff like that, which I could talk about.
I was going to say, talk about the way they can actually have the capacity to form these little homes or nests in the body. And then
so biofilms are just a great example of one of what are now just increasing numbers of cooperative strategies between microbes. The most common biofilms are bacterial biofilms. So different bacteria will begin signaling each other. And this is my best way of describing it. They theoretically can persist on their own in what's called a planktonic form. But as a single microbe, picture you're a single microbe floating around with all these other, first microbes that can outcompete you, and then the human immune system surveilling on, it makes more sense for you.
And the phages.
Yeah. And the phages and, whatever. It does not merely make sense for you to be floating around by yourself. That's a really dangerous way to be. So it seems that microbes have, bacteria have gained the capacity to form into these groups, or essentially colonies, that are called biofilms. And what's fascinating is they have all kinds of specific signaling that they have evolved to do this. So the first kind of signaling is called quorum sensing. they release these chemical messengers into the environment, and chemical messengers seem to signal others. They can be species specific, or they can reach out to a whole other range of different species, which is interesting. And they'll kind of signal like conditions, especially growth conditions. At a certain point, there's enough of a certain microbe or a certain proportion of microbes where the signal will get to a point and they begin to essentially form into a biofilm, to join together into a community. And what they do there is basically split up roles. So I think this is just key to understand about microbes, is that most microbes that persist in the body or in any ecosystem are capable of a range of activity. They can persist in a pretty innocuous state, or they can evolve and mutate in ways that allow them to act more like pathogens. Like, think about E. coli. There's, benign forms of E. coli that don't seem to do anything. And then there's E. coli that will make you, miserably sick, right? Or so they have these different capacities. And what they do a lot in order to see, how they're acting, is to alter their gene expression, which microbes can do very easily. So when they form into these communities, through this signaling, someone will say like, look, and I'm putting intent with microbes here, and there's no intent, but I'm just trying to make this simple. They'll be like, look, you take the outside, okay? Like, you turn on your genes, you turn on your defense genes, because you're on the outside of this community. So that's your job. You're protecting against, phages, the immune system, stuff like that. Now, the ones on the inside might alter their gene function so that their role is more getting nutrients and digesting nutrients, this whatever, that they will then give to the whole community, right? So there's this division of labor. People have actually, described biofilms as cities, as an analogy. Like, picture a human city where some people are doing this and that, and there's, shared processes, and we're all interacting, and we're all, communicating. It's pretty crazy, and it's definitely an incredible survival mechanism in the face of the human immune system. But recently, I thought it was fascinating, and I ended up interviewing the lead researcher who did this work. There was a team at UCSD that showed that not only do biofilm bacteria communicate with these chemical messengers that are related to quorum sensing, they actually can communicate via electrical signaling too. So he tapped into this whole energy pathways that we just didn't know existed before. They use potassium-graded ion channels to create action potentials, and literally these action potentials can propagate throughout the whole biofilm community, and they can go beyond the biofilm community. So the signals will go beyond, and they can actually recruit bacteria from distant, like really distant communities into that biofilm by communicating electrically. And
so one biofilm city can talk to the next biofilm city.
Can actually, the second study this team did showed that two different biofilm communities can actually timeshare resources. They can be in two different places. And if there's limited resources that both communities need, they will signal electrically so that one community will, begin using them at one time. They'll stop this community. They're literally coordinating to be like, look, we both need to stay alive. So how are we going to manage this? You get the nutrients now, we get the nutrients here. It's crazy, and it's so cool because it gives us so much, to me, these things are fascinating. We know that there are all these diseases of unknown cause. We know there are these conditions. We don't understand why humans do half the things that they do. And then we have these discoveries that to me are mind blowing, which is, for example, that microbes signal electrically. This signaling is pretty much the same as the signaling that human neurons use, which again leads to this whole other sort of, issue of overlap between systems. So when I was talking to Gurel Sol, who did this work, I asked him, I'm like, do you think that, signaling from microbes can affect human nerve signaling, vice versa? And he was like, well, we haven't looked at that, but I don't see why not, right? So this whole concept of a superorganism in which human and microbial cells are constantly interacting, their genes are constantly interacting, the byproducts of their metabolism are constantly interacting, is only even growing more, and that their signaling is probably interacting. I mean, we probably have all kinds of stuff going on. So when we now consider what a human being is, it's really, we are just these, mixes of microbes and all kinds of stuff that's going on.
Well, I think it also, at least to me, brings up the thought that, our bodies have receptors to sense energy.
Yeah.
That most of us don't actually notice on a day-to-day basis.
Mm-hmm.
Some people that have extreme hypersensitivity
Right.
can recognize things like electromagnetic fields
Yes.
because of these voltage-gated channels and so forth. So I think it probably is very likely that these microbiome communities may actually be communicating with us in the same kind of way.
It's definitely, in my opinion, also likely. And I actually think, when you look at treatments like acupuncture or energy-based approaches that I have personally used and have felt change the way I feel, I think it's interesting that these treatments could be tapping into these kind of networks that we have basically just begun to recognize. I think it's just another kind of, idea, like when I was first getting sick with ME/CFS, this idea that if something didn't show up on a blood test, you weren't sick. Well, now, for example, with acupuncture, we usually don't see changes on a typical blood test when someone, gets treated with acupuncture. But are we measuring the right thing? I mean, that's always a question. Do we even, I don't know if there's, any kind of energy communication you can be tapping into or addressing. We're certainly not going to pick that up on a test that doesn't even account for those pathways. So to me, it just continues to, when I look at this, but really my attitude when I look at the microbiome is that I think I just understand less as I move on. And I just not understand less, but that it becomes clear to me that this is such a complex system. The
list of questions just keeps growing.
We have actually generated more questions than we have answers. Yeah, for sure.
So, as we finish in the last few minutes of our talk together, I want you to just kind of speculate a little bit about where you see some of this going. And again, trying to tie it back into the role in terms of treating diseases like ME, autoimmune disease, and those kinds of things.
Right. Well, I'm still a big proponent of the fact that I think we need to almost just create a renewed respect for the human immune system. Over the past decades, what we've done, based a lot off the theory of autoimmunity, is create these immunosuppressive therapies. They are the top, it's a little crazy, they're the top selling drugs in the world. I actually just looked at a chart a little while ago. Humira, which, for example, just knocks down the immune system in patients with autoimmune disease, is the #1 selling drug in the world right now. And I think all the other ones under it, they're all immunosuppressants.
That would be Enbrel and
yeah.
Steroids, high-dose steroids.
They're all steroids. They all knock down different parts of the immune system. And look, they definitely help people manage symptoms. And I'm not telling anyone to stop those right now. But if we want to move forward and actually help people really get better from these illnesses, we need to understand that the immune system needs to be healthy and active in order to keep the microbiome in a state that is balanced and healthy. And these approaches that only knock down symptoms, but just literally have no regard for the microbiome, are definitely, in my opinion, only driving the increase in chronic disease that we see. The incidence of almost every chronic disease is going up right now in ways that are actually very alarming. We are living in the midst of an epidemic of chronic disease. And the fact that a huge mass amount of people in our society are immunocompromised or have their immune system shut down on a daily basis is not helping that. So what I think we need to do is and this requires paradigm shifts in thinking, and it will require a lot of paradigm shift in the research community too, is we have to begin a little more at the beginning and say, wait, how do we get the microbiome and the immune system in a place where it can, be healthy? And I think what's important with that is that we do this in the most preventative fashion as possible. So for example, when I was getting sick, back when I was 20 and I had some headaches and trouble sleeping and things, if I had a treatment at that time that had been able to help my immune system, gain back some strength, and we had some more targeted approaches that might have been able to target some of the microbes or pathogens that might have been playing a role, or a better understanding of kind of any environmental variables that were also playing into it, any, toxins, any chemicals I might have been, we would look at me as a very individualized patient, try to understand my infectious history, try to understand any exposures or negative things that might have happened to my life, understand that these can all interplay with each other towards the development of disease, that there doesn't have to be one thing causing it, that it can be a, a progressive state that moves towards a place of that's not wellness. And then we would intervene early and say, how can we bring the immune system back? How can we add balance? Maybe probiotics, maybe, bringing stuff in like that will almost prevent people from, that would be my vision. Can we intervene early in people, who show early symptoms, and can we prevent them from getting so sick in the first place? That would be the key. And I think the keys that we need to keep in mind are just, we need to respect the immune system again. We need to make immune system, strength a priority for medicine. And then we need to, again, look at very, specific antimicrobial approaches very intelligently. The use of antibiotics now, that's a whole other issue, but we use antibiotics in ways that are just not working out. We're overusing them to the point where everyone now knows we're in this crisis where a huge number of microbes are just becoming resistant to the antibiotics we use. And an antibiotic is like an atomic bomb to the microbiome right now. If you take an antibiotic, you might have, an ear infection. You get prescribed an antibiotic in the hope that antibiotic will target some microbe there. You literally decimate all your microbiome populations in an effort to see if you can get at that one pathogen. That does not make sense. We need more targeted approaches. So I think if we just keep that in mind, we can at least move in a direction that has more hope.
Amy, this has been so enlightening. I've learned a lot. And just for a teaser, for maybe in some upcoming chances that we might have to communicate with the viewers, we haven't even really touched on the role of toxic chemicals in our environment and the way that interacts with some of the microbiome. And we also haven't talked about some interventional strategies, like, for example, the use of plant-based actives, herbs, if you will, in terms of modifying, not only killing some of the bad germs when that's appropriate, but also doing some of these things to enable the immune system to do what we really want it to be doing. So I really appreciate your sharing with us kind of the sum total of all your experience and some of the ideas that you have. And I appreciate very much what you're doing with your microbe-minded series of interviews that you do. I'll have to follow those, and I hope maybe some of our viewers will seek out do you want to tell us a little bit about how people can find what you contribute, where you print and that sort of thing?
Sure. Right now, I'm just trying to put as much as I can onto my blog, which is called Microbe Minded. And I think I might even do some video blogs and some stuff that I'm going to do for that too. Right now, it's interviews and some pieces written by me that give some background. And you can also, there's links to actually published studies that I've done too that kind of, summarize scientifically the ideas. So I'm hoping there's a range of content that's a little easier to get, and then my published stuff that's more sciencey. And then also, I'm actually really active on Twitter. My Twitter handle is microbe-minded2, and so I'll usually tweet like once a day about something. And if you follow my tweets, you can get in my head.
Microbe-minded and then number 2.
Yeah, microbe-minded2. Mm-hmm. I think Twitter's pretty cool. It allows for a lot of reach and
I really look forward to seeing what you come up with, and as you start to assimilate these things. And of course, I'm in the clinic. It's what I do day to day. I see these people with ME and CFS and so forth, and we try to distill your ideas into concepts that actually help people get back into that role of wellness, and then from there it becomes prevention and a wellness program. But
that's fantastic.
Thanks so much for taking the time.
Thank you, because I love the fact that people are taking these more theoretical concepts and really trying to apply them to patients. That's key for me. And so the fact that you've been open-minded and done this is great. So thank you.
Thank you. Okay.
All right. We'll be in touch.